Hello and welcome to the Cambridge Alumni Festival, if you're just joining us. We have an all-Homerton panel for you. My name's Matthew Moss. I'm Director of External Relations and Development at Homerton College and a languages graduate, so please be gentle, you two. I'm joined by my two fellow fellows, Dr. Kenyon and Professor Ravi Gupta. Please do feel free to ask questions using the Q&A function at the bottom of your screen. But let me introduce Julia and Ravi. Julia is an independent scientist in the Department of Medicine at Cambridge. She focuses on how viral genomes are structured, and she works on HIV, FIV, the feline equivalent, dengue virus and influenza virus, and of course now the novel coronavirus, which no longer feels quite so novel. Her team develops new techniques to allow us to better visualize the viral genome. And the overall aim of her research group is to understand the process by which the virus particle forms and to design drugs that block that process. Julie is also strongly involved in teaching for Homerton as Director of Studies for Preclinical Medicine and for Biological Natural Sciences. Ravi studied medicine at Cambridge and at Oxford and went on to a master's in public health at Harvard as a Fulbright scholar. When back in the UK, he trained in infectious diseases in Oxford and in London and completed his PhD at University College London. He established a research group at UCL in 2011, working on genetics and biology of HIV resistance and reservoirs, and was promoted to full professor in 2016. In 2019, he moved back to Cambridge as Professor of Clinical Microbiology at the Cambridge Institute for Therapeutic Immunology and Infectious Diseases. He is a member of NERVTAG, the government's scarily named New and Emerging Respiratory Virus Threats Advisory Group, a meeting of which is going on as we speak, and Ravi, 2 minutes ago, was part of that meeting. In 2020, Ravi was named as one of the 100 most influential people by Time magazine, a distinction not afforded to many academics, alongside Lewis Hamilton, Phoebe Waller-Bridge, and both Joe Biden and the great Donald Trump. He was nominated by his patient, Adam Castillejo, who was known for a long time simply as the London patient, only the second person ever to be cured of HIV, thanks to the stem cell treatment that Ravi oversaw. So both with a background in HIV and both now working on COVID. It's, on the face of it, extraordinary. Homerton has 60-odd fellows across the disciplines, from architecture to zoology. So it seems really quite a coincidence that we have two fellows in virology, both working on COVID, both previously working on HIV, but it's not quite a coincidence, is it?
Well, no, because I was then in Homerton from about 2010, I think, and I was obviously aware of Ravi's research because we went to the same conferences on HIV and things, and we vaguely knew each other. And when Ravi moved to Cambridge, I was very keen that Ravi should come and join Homerton, and we managed to encourage him to do so because Homerton's quite close to the Addenbrooke site, and yeah, so it's all worked very well.
Happened. Ravi, we're delighted to have both of you in the fellowship. You've both been hugely influential in Homerton in shaping how we respond to the challenges of COVID-19. So it's great to have you with us. Let's think though about pre-COVID, if you can remember such a time. You were both working on HIV in different ways. What were your objectives, Julia, in working on HIV?
So for me, I really wanted to, and still want to, understand how the viral genome, which is a molecule called RNA, is structured. So the physical structure of this in 3 dimensions, really. Because we believe that three-dimensional structure controls a lot of different things in the viral life cycle. If we can understand how the structure changes and how we can target it with drugs, then we should be able to design future drugs against HIV and also understand the virus better. So that was the focus of my research. And in terms of RNA itself, lots of viruses use RNA similarly in their genomes, and that's why I branched out into working on several different viruses as well.
Got it. So drug development is the ultimate aim of your research, is that right? Not that you are doing that, but you're preparing.
Yeah, absolutely. We're trying to prepare the groundwork for drug development. So I was involved. Again, we don't do any of this by ourselves. We work in teams of very, very talented people, and we were trying to find molecules from already developed libraries of hundreds of thousands of compounds that might be able to target this RNA. So we were starting to do a little bit of the drug development on HIV.
Got it. Right. And Ravi, we know something of the objectives of your research because it had its ultimate expression in the curing of the London patient. But was that what you were always working towards, just developing that cure in that way?
Well, in some regards, we ended up doing cure, but really the big questions earlier on in the epidemic were all about treatment and access to treatment. And at the time I was doing my public health degree, which was sort of late 90s, antiretrovirals were just about being made available to poorer countries in Africa and Asia. There was my concern was that the public health delivery system was not very strong in many of those countries, and drug resistance was going to be a problem in my view. So we went out to kind of try and confirm or refute that, and then to understand what was actually happening. So over the next 10 years, we tracked drug resistance in people who had newly acquired HIV, as well as people on treatment who were not suppressing the virus replication in blood. And so we were able to track mutations and how they emerged, and we were able to characterize them. And that all leads to how you then bring in your second line of drugs and third line of drugs. So through using that knowledge, you improve treatment over time. And the ultimate aim is to obviously get people suppressed, because whilst the virus is out of control, you then develop, you're at risk of developing AIDS. So that's where I started, and then of course cure came along, because the Berlin patient was an example, and we had the opportunity to try and do that in a slightly different way. And
both of you then doing what seems like phenomenally important work on a very important disease for global society, and yet a coronavirus emerges from Wuhan, and suddenly drop that. Do this instead. That must have felt like quite a moment. When was it obvious that this was a moment to just sort of drop everything and work on COVID-19?
Well, I think from out we tried to resist it as long as possible, to be honest, because in virology you become an expert on one thing, and so you feel a bit uncomfortable working on other things. We were not coronavirologists, for example, so people who studied cold viruses were probably the right people to do this kind of work. But it turned out that we had to shut labs that were not working on corona. So one of these was a very pragmatic reason, was that if you didn't work on corona, you couldn't be in the lab. So that was one very strong sort of driver. Then of course we had obviously the epidemic was expanding, and we realized how serious it was. So we thought, okay, well, what can we do? And one of the first things we did was to develop to test diagnostic machines to diagnose infection as quickly as possible.
So not immediately working on vaccines, but on testing. That was your immediate focus?
That's right. That was the big question. One of the big issues was getting test results back quickly enough. And so you were having situations where people had symptoms of coronavirus, but you had to confirm it. But while they were waiting, they were mixing with other patients, and so there was lots of transmission going on within hospital. And you didn't know who had the disease and who didn't. And so the question then was, how do you separate them, and how do you place them in different parts of the hospital? So for that, you need a rapid turnaround test. And it was really critical. It's still being used to this day, the test that we validated there.
Yeah. And what was your experience, Julia?
It was a little bit different for us, because our research was very focused on RNA, so HIV, but RNA within the context of several different viruses. And so here was a virus which has this enormous RNA genome, biggest RNA genome of any virus that we we know. And we could immediately see that we could apply the techniques that we use to this virus. So we started thinking about that relatively early on. But in terms of the number of people in the lab who then contributed to the project, when it became apparent that we were going to have to close the lab unless we were doing coronavirus research, there's obviously lots of PhD students and postdocs to think about. Everybody wants to be productive in that time. Everybody wants to try and help with the cause as well. And so the entire lab shifted to this one project, which was really unusual in science and also really nice to work with everybody. So it happened quite early on, gradually, and then yeah, in April, the whole team shifted to it.
There must have been part of you, though, that thought everybody else essentially is being sent home, can't work in a lab because it's unsafe. And yet we need to carry on working in our labs. How safe are we? Were you, what conditions were you working under? Were you in hazmat suits?
No. So, I mean, the social distancing in the labs was extremely strictly enforced. So we have a huge lab which would normally have about 14 people, accommodate about 14 people, and we were working with 2 people within that lab, right, masked. So and everybody else was working from home. So I virtually never went into the lab because most of what I did could be done computationally. So we had teams working very funny hours and everybody trying to dovetail in so that we could have that very extreme social distancing but still keep going with the project.
Got it. So I have a question about vaccines, which is that we as a society have developed several vaccines against COVID-19 in what seems like absolutely record time. Those of you who've been working on other diseases like HIV must have been thinking, crikey, how have I just been really slow all of my life? If I'd just concentrated a bit harder, could we have got a vaccine for HIV by now? How is it that we end up with a choice of vaccines for COVID-19 when we still don't have one for HIV?
Well, I guess it's a question of diversity. So that's the thing. The HIV is probably the most diverse virus that we've ever known, and so the number of strains out there or different flavors of HIV is enormous, and so designing a vaccine that can hit each one is really difficult, and that's part of the problem. The other issue is, of course, getting the right levels of antibodies in the right place to defend against infection is not straightforward for HIV, and there may be an element of sort of luck in it, in that the methods we deployed worked, and some of these technologies have been used in HIV vaccine development. So part of where we are now is because of all the work that's happened on vaccines for other diseases. That's really important to recognise. Yeah.
Yeah. So these vaccines, the Oxford AstraZeneca vaccine, has been in development essentially for about 20 years because the backbone of that is a viral vector, and those groups have been working on that for a very long time. And the mRNA technology has also, so the technology behind the Pfizer jab and the Moderna vaccine, that has really come forward leaps and bounds in about the last 4 or 5 years. So if this pandemic had happened 5 years ago, we wouldn't have been in the same position with mRNA vaccines. Wow. There's had to be a lot of development in order to get that type of technology to produce a really good immune response. And we were very lucky in the timing there.
Got it. So your story of pivoting from one virus to another must have been replicated by pretty much every virologist, because every lab essentially was told, don't come in unless you're working on corona. What has that done to virology research? Have you been able to keep any of your HIV work going? And Julia, in your case, is the work that you're doing on COVID-19 going to help the HIV work that you were previously doing because of the molecule itself?
Yeah, I guess they're synergistic. So we have been able to continue the HIV work. We're back to doing HIV work now. Of course, students and postdocs, they have a project, and that project they need to publish. They want to finish, that project. They don't just want to switch to a different project halfway through what they're doing. In terms of PhD student, they can't really. They've got to produce a thesis. So the amount of sort of communal effort going into coronavirus has slightly decreased, and the original projects have increased again. But yeah, overall, everything is still able to continue, and we learn things from about each virus, really, from, well, certainly about RNA, from studying the two different ones.
Okay, and would you say, Ravi, that can you pick up your research on HIV again after this?
Yes, so we did continue a little bit of HIV work during that period. It kept it ticking along because it was using many of the same techniques. So we are planning to do that, and I think that certainly with HIV drug resistance, for example, I think that many health systems were disrupted quite severely around the world, and I think that the implications of that are not yet known, but they're likely to be quite significant.
Yeah, thank you. So, Ravi, I mentioned in the introduction that you're a member of NERVTAG. You're also a very frequent commentator in the media on COVID-19, and there's a hunger from the public for scientific facts. I guess there are journalists have you on speed dial because we can't get enough of hearing from real experts about what is actually going on. You have experience because of the London patient. You are used to explaining your work in public, but what's the comparison between the two? Is it much more intense now?
Certainly, I mean, discussing the HIV work was interesting, but it always felt a little bit niche. That some people just thought that HIV was finished, and that it wasn't an issue anymore, which is pretty much the general perception of the public at the moment. So discussing HIV was always a little bit more difficult because you had to remind people of the total burden of disease, the fact that burden was not necessarily in the UK, and that in many places the incidence of HIV was increasing, and that in some parts of the world up to 40% of adults are HIV positive. So it's sort of staggering figures like that. Of course, discussing corona has been more straightforward in terms of the questions are obvious that people are asking, and we have tried to sort of aim to answer some of them, and it's really quite satisfying to be able to do that to an audience that actually is very well informed, which has been quite remarkable. It's been really quite good fun, and also discussing things with the public or communicating is always really, really good as a scientist, because it helps you sometimes see things in a different way when you're talking about the things you're working on. So I find it really quite helpful.
Yeah. And Julia, what's been your experience of explaining to non-scientists?
So mine is much more localised, I suppose. I've been quite heavily involved in college communications and in trying to help students to understand what's happening and what might happen. I think as a part of that, one of my students told me I'm known as the person who told 1,000 students not to have sex, at which I replied, no, not true. I said, don't have sex with anyone outside your household. So, yeah. So it's been far more,
but
local.
But some people really need to hear that message. Yeah, thank you. So, I've mentioned NERVTAG, and of course there's SAGE, and there's the whole battery of scientists talking to the public about COVID-19. Some wholly independent, some on government advisory panels. Sometimes they don't wholly agree with each other. Sometimes as a member of the public who isn't immersed in this world, I feel that I have a choice of opinions available to me from the scientific establishment. So what are we to make of it when scientific experts come up with opinions that are maybe diametrically opposed, or maybe just be sort of tangential, especially when it's about our behaviour and what you're recommending that we do?
Well, I think that the discourse is part of science, and it's a good thing that the public are aware that there aren't wrong and right answers, even in an exact science such as virology or epidemiology. So I think that understanding that is helpful, because that way you frame expectations, in that the public will then not necessarily expect clear-cut answers to questions, and realise that there is uncertainty, and uncertainty is just part of the world we live in. We have to appreciate that. And then I guess often these things are resolved by discussion, and then of course political motivations come into the final sort of policy actions. So I think that it is part of a process that we're all learning about.
Yeah. What would you say?
I find your perspective on this quite interesting, because from my point of view, I used to go to conferences and hear these extremely different scientific opinions, and I think, no, I don't agree with that one, or that there's quite lively scientific conference, the way people feel about their own research. That they have very strong opinions about how many membranes are on the outside of something at a particular stage in the life cycle, or it all gets into these heated arguments. But from the perspective of COVID-19, what I've seen in terms of the scientific community is actually a very harmonious point of view compared with what I'm used to. I haven't seen anybody in the media saying something that I really disagreed with. Actually, there's my perception is that it's actually closer, and that people have more similar. I don't know how you feel, because you've got more experience in sitting on big committees. Do you find more?
I think on the committees there is more. Yeah, on the committees there is, I think, as you say, there's a more there's a narrower range of views, and I think that's because many of us have the same sort of backgrounds, and we sort of trained in very similar ways, and of course this is an unknown quantity where we're all coming from the outside, and the sort of vested interests, as it were, which often guide people's opinions, are limited.
There, are they? Yeah.
Yeah, that's interesting. So you would say that the scale of disagreements among scientists is actually much smaller because of the great focus, I suppose, than you're used to, and maybe the things they're disagreeing about are small differences to the public. It doesn't always come across like that, I think, because it's about our behaviour. Because we really want to know, if we're going to the co-op, do we put a mask on or do we not? Is this one-way system necessary? And so there is that the journey, I suppose, from the virology, what is happening in the lab, what this virus is actually doing, and the consequences for the controls that are in place in our lives, and I sense there's an element of guesswork in all of that. There's obviously the safest thing to do would be bring down the barriers, nobody moves, nobody talks to anybody else. And it's clearly a political decision rather than a virology decision, what controls you impose. But have you found that it's easy to get the scientific viewpoint across to those who are making the decisions about the social controls?
I mean, the sort of the advisory groups such as NERVTAG are only sort of there to provide advice and generate position statements or summaries, or explore areas that are requested by SAGE, for example. So they don't really have any direct policy making powers, and advice is often not sought. It's sort of just, it's left to the sort of next committee up. So, which is it's a good thing, and it's a good thing, but also I find that it does limit people's ability to influence policy, because that's taken, those decisions are taken somewhere else.
Can we look forward to a time when having the vaccine is enough, and these controls on our behaviour can be removed?
That's a difficult question. I see the pandemic going forwards as us making 2 steps forward and the virus making 1 step backwards, perhaps. I don't think there is a clear and straightforward immediate way out of this, obviously. This is going to take a little bit of time. But the number of scientific advances and the rate at which those are coming forward really should mean that, you know, the difficult periods are then getting a little bit less and less and less difficult going forward, hopefully. But obviously we do need to maintain some degree of sensible behaviour in all of this.
And of course, there are many countries in the world that don't have the vaccination rate in the population that we do, where the calculation may be different, of course. Ravi, you have experience of working overseas. When you look outside the UK, perhaps into developing countries, what sort of picture do you see?
Certainly, there are issues with vaccination. We can see that there's the problem with access, of course. And if you look at the percentages of vaccinated individuals across sub-Saharan Africa, it's very low. It's below 10% in many countries, which, you know, is shocking, really, isn't it? When we hear about the fact that, you know, vaccines are about to expire and that some are being thrown away, and the fact that we're now doing boosters in a blanket sort of fashion, it does raise some questions that really, I mean, are very reminiscent of the HIV epidemic with antiretrovirals. You know, there was, again, this big problem of lack of access to where it was needed, and it only happened through big social movements to improve access. And I think that I'd like to see more of that happening, to be honest, with vaccine equity.
So when the richer nations are sending vaccines overseas, how should we be thinking about that? Is that like food aid, in which if you provide food to a starving person, then you have helped that person, and so every little helps? Or is it like someone is stranded on a 20-story high burning building, and we turn up with a 10-foot ladder and expect that we're helping, right. But it's never going to help, actually? Is it that the goal in sharing vaccines with other nations is that enough of the global population should be vaccinated that we don't risk dangerous variations emerging? Or is it an every little helps situation? What would you say?
Well, I think limiting transmission is a key thing, and limiting the numbers of cases is key, because that's how variants arise, and that's where the big threat is going to come from. So that's the fundamental reason for vaccination in a longer-term sort of goal, which is to free up the economy, free up travel, movements between places. But you know, until we reach high levels of vaccination, there is going to be a serious problem with movement of people, I think.
But yeah, I think there's different goals to vaccination. You've got to ask, why are we vaccinating? Are we vaccinating in order to protect the individual from disease, or are we vaccinating in order to try and stop the virus from spreading? And it's much easier to do the first one. The vaccines are still extremely effective, even against the current variants, at preventing serious disease. But they are probably, and we don't have very much data about asymptomatic infection in the way that these spread. Ravi might have more than I do, but and so it's I think what we need to be doing is to be bringing forward vaccines that are more and more specific against the current strains, because we know that they were so effective against the original strain. The virus is mutating, and it's one step ahead of us, and we've got so much transmission globally that we're giving the virus so much opportunity to mutate, to create these new variants. So it's all intertwined. We need to lower the transmission rates, which we can do through vaccination, but we can also do through behaviour. And we need to be continuing to develop the vaccines so that they keep up with the virus as effectively as they can do, in my opinion anyway.
Great, thank you. Well, while we've been talking, the screen here has been filling up with questions from the audience. Let's take a few of them and fling them at you and see how many stick. So, why was the COVID pandemic not better anticipated? The WHO pandemic threat assessment was in 2005 was really only based on flu, and zoonotic diseases seemed to be on the rise, mainly owing to wildlife habitat destruction. So why has this come as such a surprise to global society?
That's a very good question. From a virology perspective, it hasn't.
Because virologists have been saying for decades that this is likely to happen.
Absolutely, yeah. The points in that question are absolutely right. The growth of cities and the encroachment into animal territories where we're bringing animals and humans into closer proximity is a breeding ground for transmission of viruses, for them to jump into a different species. So yes, we should have been prepared for that. That's probably a political thing rather than a scientific thing. And going forwards, we should now be asking, how do we prepare for the next pandemic? Where is it going to come from? Do we have the right surveillance globally to look at what's circulating in different animal populations? Because human health is really, as we've just seen, it can really be affected by what's going on in different animal populations that we hadn't predicted before.
Right. Yeah, thank you. Ravi?
Yes, I mean, we had the SARS 2003 emergency, but I think the memory, because it wasn't so catastrophic, I think the memory of that was rather short. And then MERS, again was interesting and caused some severe illness in some people, but again didn't have that impact. So I think that although these were repeated signs of what was potentially going to come, none of them were bad enough to actually, significantly change policy or research funding into those diseases.
It's interesting you bring up SARS, the original SARS, because I was doing some work out in Singapore, basically just as the pandemic began and began to hit the press. And the East Asian nations that were affected more by the original SARS tended to be better prepared for this pandemic than the nations that had not been affected personally before. So I think it probably, from just a society perspective, I think it probably takes a bit more, experience. Those societies had that experience. Their populations were extremely cautious about having another pandemic similarly, and we hadn't, in the UK, certainly in Europe, hadn't experienced that recently. So I think that might have made a bit of a difference as well.
Right, thank you. What in its research and testing for COVID-19, what should Cambridge be proud of that we can wave at our cousins on another river?
I think a number of things. For example, COG UK was orchestrated from Cambridge.
Explain.
COG UK, which is the genomics consortium that was established by Sharon Peacock, for example, who leads it currently, and COG UK did pretty much all the sequencing for SARS-CoV-2 since the beginning of the pandemic. And it leveraged labs all around the country, but was actually led from Cambridge. The other things that we've done some sort of cutting-edge work on new variants, identifying the alpha variant, for example, understanding the biological characteristics of variants, and certainly we also laid down the blueprints for how variants arise by describing an individual who had an evolving virus, and we were able to show that the virus was evolving against antibodies, and that it actually had an enhanced effect on the virus itself. So that was a sort of a blueprint for how variants emerge. And so those are the kinds of things that we've done in Cambridge.
Yeah, we've also got the asymptomatic screening programme in Cambridge, which is so important because it screens individual households of students once a week during term time. And as you can imagine, the student environment is quite conducive to transmission of this respiratory virus. And we really don't have a lot of asymptomatic data. I think I said that previously. So we've learned quite a lot about asymptomatic transmission via that University of Cambridge programme.
Fantastic. So the testing program itself then contributes to
Yep, and all of the
science.
University of Cambridge students who participate in it have been contributing to that as well. So,
fantastic. Thank you. Here's a question that we could spend another half hour on. Do you think the origin of COVID-19 will ever be definitively uncovered?
I don't think. I think it's unlikely we will get the answer, to be honest, unless we find a very close virus in the wild.
yeah, that's the only possibility, isn't it? Yeah.
And that's because a close virus in the wild would tell you that is precisely how this one happened.
Well, it would tell you that there's a direct ancestor circulating. It would argue against manipulation of a virus to get the SARS-CoV-2 that we have. But it could still have happened in a laboratory setting, for example. It could have still been, brought to a lab from the wild and then caused an infection that way. Or it could have just happened in the wild or via wet markets. So there are a number of different possibilities. But it does argue against any kind of accidental engineering of that virus.
Right. But the answer to the question seems to be no. We're unlikely ever to know how it all came about. Can any virus afford to entirely wipe out its host? Or will infectivity gradually decrease until we don't care particularly about catching COVID-19 anymore?
Most viruses, after they're at their most pathogenic usually just after they jump into a new species. So we would predict that this would be the sort of period of the virus, of viral transmission when it is most pathogenic. However, we're talking about evolutionary time here, and it could be thousands of years before the virus evolves to become a bit more used to our human hosts. And in the meantime, it will probably make humans evolve, and this is a really deadly virus for some people who catch it. So it will, viruses, they put evolutionary selective pressure on a population so that they remove the members of the population that are not resistant enough to them. Yeah, I've heard actually quite a few people saying, well, viruses get less pathogenic, so we don't need to worry about this one. But I think there is a misconception in some people that happens quickly.
Right.
It would be unlikely to happen in our lifetime, really, because this particular virus mutates very slowly in comparison with lots of other viruses. So HIV, we can see it getting a bit less pathogenic over 120 years. It's about 100 years, isn't it? But very slightly, and that is the fastest mutating virus that we know. Whereas, in terms of the RNA viruses, this one's actually the slowest.
Right. So we keep the champagne on ice for now.
I'd say so.
Yeah. Okay, thank you. Here are a couple of scientific questions for me to lob at you. Could mathematical modelling be capable of predicting variants of concern? Is it currently used for predicting variants of concern? You know?
Mathematical modelling. The issue with modelling is that we are only starting to understand what particular mutations are doing and what kind of mutations are selected by immune pressure. And we are seeing unexpected mutations that enhance, for example, particle or virus infectivity or how well the virus binds to cells. And so I don't think currently mathematical modelling will have the input data required to make accurate predictions. And of course, we know most about spike. We know very little about all the other proteins in the virus. And so that, for modelling to work, you need much better underlying knowledge, I think.
I think that's right. We need to understand the molecular biology better in order to fit the modelling into it. But we did a little bit of work on that with RNA and the RNA structure of the virus, and saw that the mutations are cropping up in particular regions. So there's a bit of a pattern to where they're cropping up. So it might well be that looking into things like this more would allow us to predict better what's going to turn up. I don't think we're ever going to be able to predict fully. But
Right. Yeah. Okay. So maybe never, but certainly not yet. second scientific question, one for Julia maybe. What is the mechanism of mRNA versus DNA vaccines?
So our genetic code is obviously DNA, and that genetic code has to become protein in our cells. Proteins are the doing molecules, and DNA is the molecule that encodes all of the genetic information. mRNA is the messenger between the two. It's part genetic code and it's part functional as well. So in a DNA vaccine, we inject DNA into somebody, that goes into their cells, and it becomes RNA, which becomes protein. With an mRNA vaccine, you inject the mRNA, and it just has to go from mRNA to protein. So they're both genetic forms of vaccine. We're introducing the viruses or the antigen that we want to protect against. We introduce that genetically rather than as a protein. There are also vaccines available that are just protein.
Right, thank you. Here's a question from someone called John Bell. Don't want to scare you. Global pandemics have occurred frequently, but eventually decline to much lower levels. Without mass vaccination in many cases, is COVID-19 any different? And might it not eventually mutate to a less damaging predominant variant, particularly with the help of vaccines?
That's an odd question. Well, vaccines will change the trajectory of evolution, there's no doubt. But in my view, from what we are seeing with the Delta variant, for example, one way that the virus is getting around vaccines is not simply to change the way that it's recognized by antibodies from vaccines, but also just to become hotter. In other words, replicate faster and infect quicker and generate more particles. So that's really quite worrying, because that tells you that the virus has different strategies, and the byproduct of making more virus means that you get more inflammation, and therefore data suggesting you're twice as likely to be hospitalized with Delta as compared to previous variants. So if anything, this virus is getting more pathogenic. We had the Wuhan, then we had Alpha, which showed a potentially increase in mortality with infections with that variant, and Delta probably is the same. Again, an incremental increase. So in fact, we're going the opposite direction.
Yeah,
Right.
We haven't been able to follow any of the other pandemics through history in the same way that we can do this one, in terms of, from a molecular perspective, what is the circulating predominant variant, and what's the pathology of that. So, as Ravi said, this one has just got worse, really, rather than better. And although across a very long evolutionary time, you would expect that, hopefully, to calm down and get better, because there are lots of viruses that don't affect humans at all, really, or are beneficial to humans even. But that might, as we said before, take a very long time.
Right. Thank you. Now, there's a couple of questions on vaccine hesitancy. How do you best counter the anti-vax movement? And I guess there's a spectrum of reasons why people might be hesitant to take the vaccine. How would you suggest that you can best explain, or, well, maybe explaining actually isn't going to be useful for some of the people who are militantly anti-vax, but do you have any recommendations for how we get the vaccine rate up and how we reach that particularly reluctant section of the population?
I mean, my view is that to use history and to show people what the world was like without vaccines. In other words, pictures of people dying from smallpox or other lethal diseases, and then to say, well, we eliminated one of those deadly pathogens. We now have vaccines for many others that have actually probably been less rigorously tested than the SARS vaccines. The amount of rigor in the SARS vaccinations trials is huge. And so, if anything, there's much more data on safety of this vaccine than there was for the other vaccines when they were rolled out into practice. So that's why I find it really confusing as to why there is a hesitancy to start with, unless those people have refused all vaccines. in which case that's a consistent argument that for whatever reason people are not, don't want to be vaccinated. But to single out coronavirus vaccines against all others, I think is just not logical, and I'm not sure how I would approach that.
Do you have any magic bullet for this, Julie?
No, I think that's more of a behavioural scientist area, but I guess trying to understand the reasoning behind it is really important, because if you have the educational background to understand what vaccines do, then you would be very unlikely to refuse this vaccine. And also to understand that there is risk associated with everything that we do, and that the risks of vaccination are absolutely tiny in compared with the risks, when we compare to the risks of disease from this virus in particular. And we don't actually know the long-term effects of this virus really going forward, 5 years, 10 years. We can't measure that yet, and I find that really quite concerning, given the number of cell types that the virus will infect, and the number of different symptoms that it causes in patients, which is really quite big.
Does the calculation about the safety of the COVID-19 virus, Ravi, does that apply as we extend the sections of the population who are getting it? So is it less safe to vaccinate children, for example? Or can we simply be confident that the measures in place and the data that's in place on the safety of the vaccine should give us confidence that this is all fine?
Well, we have waited sort of for vaccinating children, and we've now gathered data from other countries who did it earlier. So by waiting, the data now available for the incidence of various side effects from the vaccine in children, and they appear to be very rare. So that's reassuring. So I think that sort of having a childhood vaccination program is certainly, in my view, important.
And should we be vaccinating every last person in the UK, or should we be focusing instead on vaccinating those in sub-Saharan Africa, as you say?
It's and again, difficult question. I think that doing both should be feasible if we can get production scaled up. One of the big blocks has been licensing agreements from some of the companies who have sort of dragged their heels, and really governments have not really stepped up to deal with this, and in some cases have supported those blockades. So I think that this is a political barrier, not a sort of production-related issue.
So it's not how many vaccines there are in the world, it's where those vaccines are, and how we can better distribute them. Yeah, thank you. I have a final question on policy again. The policy in New Zealand contrasts with the policy that we've seen elsewhere. There's been very strong lockdown and zero tolerance of COVID. Is that a policy that can last? Is it possible for New Zealand to keep its barriers up until all of this goes away?
I think they'd be doing that for a very long time if they were to. I don't think this is going to go away immediately. But I would say I know that the populations of those countries, Australia and New Zealand in particular, they've really made a lot of sacrifices in terms of being able to go overseas, see relatives, travel. And the lack of spread in those countries is good for all of us. So, thank you, Australia and New Zealand and all other countries who control that well.
Great, thank you. I think that's the final question that we have time for, but Ravi, Julia, thank you so much. I've learnt a heck of a lot. I was an undergraduate in this lecture theatre 100 years ago when I learnt a tremendous amount, and I feel I've just done it again. So thank you hugely, and thank you to the organisers of the Cambridge Alumni Festival for having us here, and I hope our audience watching at home enjoy the rest of the show. Thank you.